everyone. Here we are with reproduction, which is the very last of the lectures. So, there's that. The other thing is reproduction is my favorite lecture. So I'm going to have fun going through this. Just going to, first off, let's show off my reproduction here. Here's my daughter and my son. Okay, so let's get started. Starting with, well, what is the point of all of this reproduction? Okay, so remember that the reason we go through that whole shenanigan of sex and all of our offspring and all of that is to create diversity within the population so that the offspring are not going to be the same as the parents or anybody else who's ever been. Okay, so it's all about creating unique offspring that are different from everybody else because diversity makes us stronger. We've already talked about this before. The other thing that's important is in the process, you need to make sure that the offspring have the same number of chromosomes as the parents. Okay, so we are all diploid. What does that mean? Diploid, di, meaning two, it means that we have two sets of each of those chromosomes. We've got 23 chromosomes and we have two copies of each, except for males. That 23rd chromosome is a sex chromosome and males have an X and a Y instead of two X's. But other than that, we all have two sets of each chromosome. So we are diploid. So when you see this notation right here, 2N, N is referring to the number of chromosomes, and 2 refers to twice as many. So we have twice as many chromosomes, 23 chromosomes times 2. Okay, so we are diploid. So the way that this is going to work is the males are going to produce a haploid, hap meaning half of, and half of 2 is going to be 1. So the males produce a gamete and the females produce a gamete, and then they're going to come together. And when you have two haploid cells, so an egg and the sperm, they're going to come together to create, again, a diploid zygote, which will then develop into an embryo and then to a fetus, and then eventually give birth to another diploid offspring. Okay, so you have to have half as many chromosomes so that when you combine together, and again, the combination, diversity, that's the whole point, recombining it back together, and that brings you back up to the same number of chromosomes that you started with from the adult parent. Okay, let's go to the males first. The reason we start with the males is because they tend to have a more simplified system, so we're going to go through that first. Okay, so the first thing I wanted to do is do a little bit of an anatomy review because I know not all of us remember our anatomy quite the same. So let me just go through really quick some of the anatomy, and then we can talk a little bit more specifically about the physiology of it. So here is the testes, and the testes is a mixed gland. That would be the gonad for males. And as a mixed gland, that means that it has both an endocrine product and an exocrine product. So what would be the exocrine product of the testes? That would be the sperm. Okay, so we create the exocrine product, which would be the sperm, because it'll be secreted onto a surface and come out of the body. So that the sperm would be the exocrine product. And then the endocrine product would be androgens, and especially testosterone. Okay, so that'll be produced in the testes. From there, the sperm will be transferred to the epididymis. There's the epididymis. To the ductus deferens, also known as a vas deferens. That's what's going to be cut in a vasectomy. So the ductus deferens, which loop over the ureters and then empty into the urethra. And then from there, the urethra will transport these sperm out through the penis. Okay, so that's the basic general ductwork. One thing that's important to understand is that both the endocrine and the exocrine products, so both the sperm and the testosterone, are going to be produced and secreted in continuous, constant levels. Okay, so going to be held at a fairly consistent level through adulthood, from puberty through most of adulthood. Okay, that's not going to be the case for women. Okay, so let's look at a little bit inside of the testes. So here's the testes right here. If you look inside of the testes, there's going to be a whole bunch of little coils. These coils are called seminiferous tubules, and the seminiferous tubules, there's the word right there, that is going to be the location of where the sperm are created. And lining the lumen of the seminiferous tubules are stem cells. Okay, so if you're looking at this picture, here's a cross section through the seminiferous tubule. Here is a stem cell, and these stem cells will undergo meiosis to produce the sperm there. We're about to go there in a second. And so you'll notice that as we go towards the lumen, so towards the inside, the empty space of the seminiferous tubules, you get a more and more developed sperm. Okay, so as you go towards the middle, you have more and more developed sperm until the point where you get all the way to the middle. You can even see their little tails sticking out into the lumen there. Okay, so the other thing that you'll notice is between the seminiferous tubules, you have a different type of cell. Those are called Leydig cells. Leydig cells. And the Leydig cells, they produce something different. They are the exocrine part, and so they're going to be producing the testosterone. So we have sperm production inside the seminiferous tubules. They're sort of helped by some supporting cells, which are called Sertoli cells. And then we have Leydig cells, which are kind of between the cells, and they produce testosterone. So here we have spermatogenesis, and spermatogenesis is just the production of sperm. And the good news is, for males, spermatogenesis is just the same exact meiosis that you already saw at the beginning of the semester. Okay, so when we learned about meiosis at the beginning of the semester, what we were really learning about is spermatogenesis, because when we talk about females, oh, genesis, there's some other changes. Okay, so it's the same process you already learned, but let's go through it a little bit together. The first is you always start with your stem cells. Here are your stem cells right there, and those stem cells are going to make more stem cells by doing mitosis. That's what this is here. So this is mitosis, that's producing identical cells, just making more stem cells. But when they start doing meiosis, remember that there are two divisions in meiosis, and they're called meiosis 1 and meiosis 2. And each one of these, meiosis 1 and meiosis 2, each one of them has a prophase, metaphase, anaphase, and telophase. And that's why I sometimes like to say PMAT times 2. That's just how I remember it. It's nothing official. Okay, so prophase, metaphase, anaphase, telophase 1, and then prophase 2, prophase, and so forth. Okay, so two divisions stacked on top of each other with no replication in between. Okay, so here's the first thing we have. Before meiosis 1 occurs, before we can start dividing, we need to replicate the DNA, which just means you take the DNA and you make extra copies. Now, usually they still stay with the 2N, but in reality, how many copies are you going to have? So if you start with 2 copies, so here's the maternal chromosome, here's the paternal chromosome. Before you divide, you need to replicate, which means that you're going to replicate the maternal. Here, now we have 2 maternal chromosomes, and replicate the paternal. Now we have 2 paternal chromosomes. So these are usually still designated as 2N because here's maternal, here's paternal, but really how many of each do you have? 1, 2, 3, 4. That is 4 sets of that chromosome. So for example, if this is chromosome 13, I don't know, I just chose one. So chromosome 13, I now have 4 chromosome 13s, okay? So you'll have twice as many, so it's really more like 4N. And then during meiosis 1, you'll have prophase, where they get ready to divide, metaphase, where they line up, anaphase, where they separate, and then telophase, okay? And then that's the end of meiosis 1. At this point here, they divided. You'll have 2 cells here, Okay, so 2 cells, 1, 2, but you still need to divide again. So what then is going to happen is then you'll divide those apart and those apart, and so you'll end up with 1, 2, 3, 4 different cells, and each one of those cells has half as many, or one set of each chromosome. Now, the mixture of maternal versus paternal is really going to depend upon luck, chance, and so that's called independent assortment, and that helps increase the uniqueness, the diversity within the gametes that are produced. Okay, so you'll have 1, 2, 3, 4 viable sperm from one meiosis process. Okay, so keep that in mind for later. So 2 stacked divisions, PMAT times 2, and 4 viable sperm coming at the other end. Each one is going to be half haploid, half as many, and they're all going to be special and unique from each of the others. Okay, now let's talk about hormonal control. So as always, often the story starts at the hypothalamus. Okay, it's the king. So the hypothalamus, it's going to secrete a releasing hormone. Anytime you see RH, that stands for releasing hormone. And so it'll release gonadotropin. So GN stands for gonadotropin, releasing hormone. That then goes to the anterior pituitary, which is the master gland, because it releases tropic hormones. And the tropic hormones, specifically with the gonadotropins, those are going to be follicle-stimulating hormone and luteinizing hormone. Now, the follicle-stimulating hormone and the luteinizing hormone, they're both going to go to the testes. So they'll be secreted into the bloodstream, travel around until they reach the testes. And once they reach the testes, they're going to have different effects on the testes. Okay, so the first one, the follicle-stimulating hormone, it's going to go to the Sertoli cells, which I remember I said that the Sertoli cells are the supporting cells that help with spermatogenesis. So follicle-stimulating hormone is going to stimulate spermatogenesis. The other thing is that those Sertoli cells secrete a hormone that it then goes back and inhibits the anterior pituitary. Okay, so it'll reduce the production of follicle-stimulating hormone, which again kind of makes that negative feedback loop that we already saw. Okay, so remember the relationship. Follicle-stimulating hormone stimulates spermatogenesis and inhibition, which in turn inhibits itself, and that helps keep a fairly consistent level of spermatogenesis through adult life. And then the other one, luteinizing hormone, and the names for these will make much more sense when we get to females. Okay, they're named based on what they do in the females. The luteinizing hormone then is going to stimulate the Leydig cells. Remember, the Leydig cells, those are the endocrine regions of the testes between the seminiferous tubules there. And they're going to stimulate the production of testosterone. And then the testosterone will then have a negative feedback on both the anterior pituitary and the hypothalamus, thereby controlling the levels of testosterone. So again, the levels of testosterone should stay fairly consistent throughout adult life after puberty. Okay, so let's see what happens at puberty. So one thing I'd like you to notice is, except for when they're fetus and they're really, really newborn, most of the childhood, so before puberty, you're going to have very, very low levels of testosterone, very, very low levels of testosterone. And the reason for that is because the hypothalamus is very sensitive to testosterone during those years. And so that even those very low levels of testosterone is going to result in a low level of gonadotropin releasing hormone, and then the low level of FSH and LH. Okay, so that helps keep the testosterone levels really low. And so what happens during puberty? During puberty, the hypothalamus loses some of that sensitivity to the testosterone. So if the hypothalamus is less sensitive to testosterone, that's going to result in an increase in gonadotropin-releasing hormone, which would result in an increase of both of the gonadotropins, FSH and LH. And that increase in LH is going to result in an increase in testosterone. So basically what's happening here is it's just resetting the threshold. That's really what it is. Okay, so you're still doing the negative feedback loop, but you've reset the threshold so that the testosterone levels are going to be maintained at a higher level. So after the fluctuation and the increase that happens during puberty kind of levels out, from then on through the most of the rest of the adult level, it's going to stay at a fairly consistent level. Okay, so you have, a fairly consistent level of testosterone for the rest of most of the adult life until you reach older ages. Then you can sometimes have a decline in testosterone. The other thing that I wanted to notice is, see, look at this blue line. So this blue line is, here we have the sperm production, and the reason it doesn't start until puberty is because the levels of the follicle stimulating hormone are too low to stimulate the Sertoli cells, which means that you're not producing the sperm. So you're not going to be producing any sperm until puberty. And at that point, because of the changes that happen in the hypothalamus, where it becomes less sensitive to testosterone, that's when you're going to have the increase in follicle stimulating hormone. And as the levels of the follicle stimulating hormone increases, then your levels of sperm production will also increase. And notice that once they've been reset to adult levels, the sperm production stays fairly consistent, again, throughout most of their life. Okay, so men can do produce sperm for long periods of time in their life, well into, advanced age. Okay, so that's one major difference between males and females. Okay, let's go into a little bit about the effects of testosterone, and we've all been through puberty, so we all generally know what this happens here. So testosterone it's a steroid, and as a steroid, it's a lipid-soluble hormone. And remember, for lipid-soluble hormones, their receptors are inside of the cells, target cells, and even more importantly than that, they travel to the nucleus and they change gene expression. So they're going to be producing entirely new proteins, and because they're changing the expression patterns for the genes, that means that they could have fairly, big effects on the body. Okay, so they change primary sexual characteristics, things like growing the gonads, growing the penis, all of those sexual organs, starting up that spermatogenesis, but they also have other, kind of widespread effects on the body, broader shoulders, changes in the skeleton, changes in the muscle mass, changes in body hair, so your epidermis, your dermis is changing, changes in how mood and aggressiveness, and just like all of these different effects. So big effects as steroid lipid soluble hormones. Okay, so a little mini review here. Which one stimulates spermatogenesis? Okay, so that's going to be follicle stimulating hormone, and the way that it works is by stimulating those Sertoli cells, which then help support and make it possible for the sperm to undergo spermatogenesis. Which one stimulates testosterone production? That one's going to be luteinizing hormone by stimulating the Leydig cells to produce testosterone. Which one's going to be released by the hypothalamus? Well, if it's from the hypothalamus, it's got to have an RH in it, releasing hormone. So that's gonadotropin releasing hormone. And which one stimulates primary and secondary characteristics? Well, that would be testosterone. Okay, so that's enough for now. I'm going to come back and do another one for females, and then another one for pregnancy and menopause.